Good morning, good afternoon, and good evening. It is Tuesday, March 3rd, 2020. My name is Wei Su from Biopharmaceutics Group in Merck Research Laboratories. On behalf of GastroPlus user group, welcome everyone to today's webinar. Presenting this morning will be Amitava Mitra and Neil Parrott. An opportunity to ask questions will follow today's presentation. You may send your written questions using the questions pane on your control panel, or you may use the hand-raising icon on your control panel to ask your question directly. Please be sure to enter the unique audio pin displayed when you join the call. This webinar is being recorded for playback on www.simulations-plus.com. Before we get started with the presentation, we have a quick poll question. So the poll question is that, how many years have you been using GastroPlus? And you have 30 seconds to answer the question. All right, looks like the pool is closed and we have our polling results here. So it looks like the majority of the folks here on the call have less than 1 year of experiences. So I would say certainly this is a good opportunity for the audience, to listen to the topic with the two experts who are presenting today. And of course, there are folks with more years of experiences, and I think it's also a great time for some interactions and in the Q&A answers as well for this specific topic. All right, so now it is my pleasure to introduce our speakers today. Our first speaker, Amitava Mitra, is a scientific director in clinical pharmacology and pharmacokinetics group in Janssen R&D. He has previously worked in clinical development at Sandoz, which is a Novartis division, and prior to that in biopharmaceutics at Merck and Company. Amitava has a PhD in pharmaceutical sciences from University of Maryland, Baltimore. Amitava's main research interests include clinical pharmacology, modeling and simulations, pharmacokinetics, and biopharmaceutics. He has interacted with FDA and EMA on these topics as part of project support, as well as in workshops and consortia. Amitava has published more than 40 research and review articles and had 30 podium presentations in national and international conferences on these topics. Amitava has also involved in several cross-industry consortia such as IMI on oral biopharmaceutics tours and IQ. He's also active in ATS and ASCPT, and he was the Clinical Pharmacology Track Chair on 2019 APS PharmSci 360 Scientific Programming Committee. Our second speaker, Neil Parrott, is a distinguished scientist in the Translational Modeling and Simulation Group, which is part of the Pharmaceutical Sciences Department of Roche Pharmaceutical Research and Early Development in Basel, Switzerland. Neil specialized in physiologically based pharmacokinetic modeling and its application to Roche projects from early discovery to the market. Neil has a particular interest in the application of PBBM modeling for first-in-human PK projection and to guide formulation development and the pediatric drug development. He's involved in research activities to develop better PBPK models and is also active in cross-industry consortia. Neil has published over 100 papers and has given numerous presentations at international conferences. Now, without further ado, I'll turn over to Amitabh for this webinar.
Thank you, A. Appreciate the kind introduction. On behalf of Neil and myself, I welcome everybody. Thank you for your time today, and hopefully this will be a worthwhile time that you spend, and we're looking forward to some interesting discussions and Q&A at the end of this presentation. So Neil, next slide, please. All right. So here's an overview of the presentation. So Neil and I are going to do a tag team presentation, as we just alluded to. So I will go over the background of pH-dependent DDI and briefly introduce the paper that we just published across industry. Neil, myself, and co-authors from few other pharma companies. And then Neil is going to do a deep dive on the case studies and future outlook, and then end it from there, and then we'll go into a Q&A after that. Next slide, please, Neil. So as a background, again, I mean, we all know that PK-related DDIs can cause toxicity or poor efficacy for drugs, and which needs to be avoided as much as possible, because these AEs can contribute to significant patient harm and overall cost of development, also increase the cost to healthcare in general. And by and large, PBPK over the last few years have been instrumental in predicting and managing metabolic DDIs. And also there are numerous examples now where PBPK has been helpful in labeling and label languages across regulatory agencies. Now, where we feel there is still work to be done and room for improvement is application of PBPK or PBBM, which is probably physiologically based biopharmaceutics modeling, however you want to call it. There is significant room that PBPK or PBBM can play in the area of absorption-related DDIs, and that's what we're going to focus on today, primarily in the area of pH-dependent DDI when a drug is taken with an acid-reducing agent. Next slide, please, Neil. So before we talk about the effect of what this acid-reducing agent can have on a PK of a drug, so just so we get the nomenclatures right, ARA or acid-reducing agent is a broad term that is used to define proton pump inhibitors, H2 receptor antagonists, and antacids. And sometimes these terminologies are used interchangeably, but there are distinct differences in the mechanism of action of a PPI versus the H2 receptor antagonist versus an antacid. All right. So again, we are not going to go into those details here, but for the audience, it might be obvious to most of you, if not all of you, but I just wanted to make sure that these terms are that everybody understands that PPI, H2 receptor antagonists are kind of used interchangeably, but there are distinct differences in their mechanism of action, effects on pH, transit times, and things like that. So the question is, how would ARA or an acid-reducing agent affect the PK of a drug? And now the figure on the left that's shown here is a pH solubility profile of a weekly basic drug. And it's a pretty typical profile. The profile might be a little different. Slopes might be different and such, but depending on the pKa of the drug. But it's a pretty typical profile of a weekly basic drug where you see fairly high solubility in the lower pHs, and then solubility drops as the pH increases, again, depending on the pKa of the drug. So what you might imagine would happen is if these drugs are taken with a drug which would increase the pH of the stomach, then the solubility of the drug or the dissolution of the drug product in the stomach would decrease. Either the extent of dissolution or extent of solubility or the rate of solubilization, either or both can decrease, and sometimes significantly. So what that would mean eventually is that would affect the absorption of the drug once it transits from the stomach to the small intestine and beyond, and that would of course have an effect on PK. Now, to illustrate that, if you hit enter, Neil, one more time, please. So here is a parameter sensitivity analysis just to illustrate that effect. So what you see here is a three-dimensional PSA. The y-axis is the fraction absorbed of a given drug. The x-axis is the stomach pH, and the z-axis is the dose. Now, what is meant to show here is, again, this is a weekly basic drug with a steep pH solubility profile, pretty similar to the profile that you see on the left, the solubility profile. Now, as you can see, there's significant impact on fraction absorbed of this drug, which is the y-axis, as the stomach pH increases and the dose increases. So much so that if, as the dose increases to above 500 milligrams at a higher stomach pH range of around 5 or so, the fraction absorbed is in the teens. All right. So you can imagine in a scenario where it is a first-in-human single-dose escalation study being conducted, and it is a weekly basic drug, something like this, and as you're escalating dose, at some point of time, for a drug like this, there is going to be a saturation of exposure unless it is somehow mitigated with the right kind of formulation, et cetera, et cetera. So this kind of parameter sensitivity analysis could be extremely helpful to understand how the drug is going to behave on dose escalation and the risks of dose escalations, especially in before a first-in-human study is conducted, and the risk is appropriately communicated to the clinical pharmacologist or the clinicians on the team, so that there is no nasty surprise in a first-in-human study like that. Next slide, please, Neil. So, and there are real implications. This is not hypothetical at all. There are many publications on the effect of ARA on week basis, and we're just going to show you some of the highlights here. So this figure here shows the effect of ranitidine, which is the H2 receptor antagonist, on the PK of ketoconazole. So as you can see, the exposure of ketoconazole decreases 95% when it's given with ranitidine. Next, please, Neil. Thank you. And this is an example of atazanavir. Again, it's an antiviral. When given with a PPI, proton pump inhibitor, lansoprazole, again, approximately 95% decrease in exposure. And it is not just an effect on PK itself. This can have significant effect, obviously, on the efficacy of these products, because if there's again, in this particular case, if there's a 95% decrease in exposure, you would assume that it's going to have pretty significant effect on the efficacy of these drugs too. And something like atazanavir, which is an antiretroviral for HIV, it can have pretty significant effect on the patient population. If you could go to the next slide, please, Neil. So here's an example of how that affects the label. And this is, again, atazanavir, which I mentioned is an antiviral for HIV. If you look at the label of this product, this is the US label. In the dosing recommendation, and I've highlighted one sentence there, which clearly mentions that when co-administered with either a H2 receptor antagonist or a PPI, dose separation may be required. And the label goes into much more detail on when this is needed. Next, please. And here you go. So again, highlighted there. I don't want to read verbatim what says there, but again, you can pull up the label. The link is provided at the bottom of the slide here. But what it says is, in treatment-naive patient, there is a maximum dose of a PPI that can be given, and it needs to be separated by approximately 12 hours between the PPI and atazanavir dose. And in treatment-experienced patients, effect of use of PPI is contraindicated. So this just gives you a sense of the effect of these kind of DDIs on the label. And also you can have to think about it from a commercial perspective, because the environment that we are in right now, multiple companies sometimes work on the same target, and if you end up with a label which has restrictions like this versus a competitor which does not have label restrictions, obviously they will have a much more commercially viable product than a label with a restriction like this. So something to keep in mind. So it's beyond science, if you may. Next slide, please, Neil. So obviously the agencies have taken a note of this, and what we're showing you here is a public docket that FDA published in 2018, asking for comments from the public, especially on a framework of the assessments that need to be done for pH-dependent DDI, and it's a pretty broad ask. They're looking for inputs on in vitro studies, clinical studies, preclinical assessments, and also modeling and simulation. And this docket was one of the impetus that led us to work on this cross industry manuscript that we're going to talk about, just showing our agency colleagues, both FDA, EMA, and others, on how we feel PBPK or PBBM can be extremely useful in assessing the effects of ARAs on PK. Next slide, please, Neil. So again, we don't want to obviously mean to say that this is the only paper that has been published. Not at all. There are many publications in this area. I've just given you some highlights here. This is not a laundry list. There are many publications, but this just gives you a snapshot of the kind of work that has been conducted in the ARA area and the publications thereof. So it goes from preclinical assessments to studying development of formulations. There's a nice paper from Novartis a year or so back on de-risking discovery compounds. There are investigations of bioequivalence and some basic research, if you may, i.e. characterizing the GI contents when given with acid-reducing agent, proposal of biorelevant media, understanding the variability in the physiology and transit times and such when given with when subjects take ARAs. And there are many publications out there, so you can easily do a PubMed search and bring up a lot of those if you're interested. Next slide, please, Neil. So here is the paper that we published. It is now published. It's available online, and I think it is open access for at least some period of time, if I'm not mistaken. So it's in JPS. It's available online, as I just mentioned. So what we have here are 7 case studies of insoluble weak-base compounds, and these are examples from 5 pharma companies, which includes Roche, Novartis, Janssen, Merck, and GSK. And the authors obviously listed there. So we think it provides a nice overview of the collective experience of these 5 companies. And also we have a pretty thorough list of other publications that are in this area in the supplementary materials which accompanies this paper. Next slide, please, Neil. So here's a snapshot. Again, I would not go into the details, but this table is from the paper. So if you are interested, if you have not looked at the paper yet, I will obviously highly encourage you to look at the paper. So this table is on the paper. So this gives a highlight of the seven compounds that we have used. As I mentioned, these are all weak bases, which is obviously the point of this paper. All BCS2 and 4 compounds, and we have quite a broad range of doses across these case studies, as you can see, which is the 5th row or so from the top, from 50 milligrams to up to a gram dose. That I mean, so it gives you an overview of the effect of doses that can have on the PK. And I would particularly highlight that in all of these cases, we had a pretty robust pH solubility profile measured data that was available, which is one of the key inputs for any of this kind of modeling. So that's something I would definitely highlight it as you work on your project, is to have a pretty robust, accurate pH solubility or biorelevant solubility data set. Next slide, please, Neil. So based on our collective experience, what we have also done is provided a workflow, at least our thinking on how one can go about modeling effects of ARA. And again, this is in the paper, so I will not go into too much detail, and I don't expect you to read too much in here right now, but again, this is all clearly laid out in the paper. So there are some assumptions or ground rules, if you may, that we have to keep in mind before we venture into this kind of PBBM modeling for ARAs. The first assumption is the mechanism of the effect of the ARA on PK is primarily solubility based. That's the effect of pH on solubility. So there is no chelation between the ARA. There is no metabolic DDI between the API and the ARA. And the reason why we're calling it out is because that's also known. There are publications that these kind of interactions might happen, but the modeling that we are going to talk about would not necessarily capture that. It is always nice to have clinical data to verify or validate the model, at least maybe even a first-in-human single ascending dose data in healthy volunteers. And again, most of the case studies here are free form of the API in a dosage form. This does not mean to say that we can't model salts or effects of an acidifier in the formulation, but that would mean that you need to have other inputs beyond just solubility. For example, dissolution, and there needs to be a pretty robust dissolution experiments that need to be done. If you go one more, Neil, please. So in the pre-FIH state, what we would typically do is, because there is no clinical data to verify, I think where this is most useful is a PSA, a parameter sensitivity analysis based on the physicochemical properties of the drug that I'd shown a few slides back. I think that's where it kind of helps to highlight the risks, if any, of dose escalation on PK. One more, Neil, please. Now, well, once you have, let's say, some FIH data to verify or validate the model, I mean, that's why you would go into developing a full-fledged PBPK model, if you may, trying to predict the whole PK profile and the PK parameters and verify that vis-a-vis a first-in-human data, hopefully in healthy volunteers. And if you have IV data, that's even better, which sometimes you may or may not have, depending on the strategy of every company. So, and again, we would mostly suggest that, you know, use the default physiology as much as possible, unless there is a very good reasoning and rationale to change the physiology to fit the data. Next slide, please, Neil. Next one. And to predict the ARA, of course, once you have built and verified the model based on available clinical data, then you go about predicting the effect of ARA, which would primarily be changing the pH in the stomach range, and depending on whether it's a PPI or H2 receptor antagonist or an antacid that you plan to predict, the pHs can vary. Typically, it's around 4 to 5 in that range. Again, a PS effect of any changes in physiology or any other physical properties, that studying that by doing a parameter sensitivity analysis is extremely helpful. Next, one more, please, Neil. And eventually, you know, once this model is developed and verified, I mean, sometimes if again the strategy of the ClinPharm program, if there is effect of ARA which has been studied either in a phase 1 either in a first-in-human setting or outside of a first-in-human, that data is always very nice to have to verify the model. And if the model does in fact predict that there is going to be significant clinically significant. I think that's an important delineation here. Clinically significant PK effect of the ARA, then these models can be used to help in formulation development. But if there is no effect that's predicted, or not clinically relevant effect that's predicted, then the model can be used not only to obviously help the clinical program, but also for informing the label. And Neil is going to have a case study talking about that. So with that, I am done, and I will hand over the baton to Neil.
Thanks, Amitava. So as we move to the second portion of the webinar, we have another quick poll questions for the audience. So the question is, have you used gas plots to model the effect of acid-reducing agents? Please select one of the followings, and you have, again, 30 seconds to respond. All right, so the poll is closed. Looks like majority of the audience has not used special plus for the modeling effect of the ARA. And if yes, it is mostly for internal division support rather than for regulatory interactions. All right, so with this in mind, so we're moving forward to the second phase of the presentation by Neil.
Hi, everybody, and thanks very much, Amitava, for that really nice overview of the paper. And it's now my privilege to take up the second part of this presentation, going into some of these case studies in a little more detail and building on what Amitava has already presented. So before I start on this first case study, I'd just like to again highlight that the paper comes with a rather substantial document of supplementary material, which includes more details about the specific case studies covered in the paper, but also within that supplement is a table which contains a list of publications which have been published on this topic. And so please do, if you're interested, go in and have a look at that supplement, and you will find there links to many of the publications in this area. And in fact, my first case study is one of my own papers on the molecule alectinib. This is a Roche oncology medicine for non-small cell lung cancer. And this was published back in 2016. So to take you through alectinib's properties, as you can see from the table, it's a lipophilic basic molecule. Permeability is good for alectinib. But if you look at the chart on the right-hand side of the slide, you'll see that the solubility in aqueous media is very low. So at the low pH of around 2, solubility is only about 10 microgram per milliliter. And then as pH increases above 3 to about 6, then you see a very rapid fall off in solubility to extremely low values. However, going back to the table on the other side of the slide, you see that when measured in biorelevant media, in a physiological temperature, and in this small scale dissolution kinetic solubility assay, the actual solubility is greatly enhanced in the biorelevant media. And those biorelevant values were used in building up the model. The last thing to mention from this slide is that the clinical dose of alectinib is rather high, especially when you consider that in the context of this very low solubility. So the model building process won't be covered by me here, although you can find that in the publication. Basically, we had intravenous data with which to describe the disposition and use that in GastroPlus as a compartmental model. And we also had additional data with a suspension formulation in order to verify the oral absorption model. What I want to focus on with this slide is how we modified the GastroPlus ACAT model to describe the effect of the proton pump inhibitors. And you see, in the fasted state, normal gastric pH is about 1.3. We increased that to a value of 4.5 to simulate the effect of the PPI in fasted state. Alectinib was recommended to be dosed in the fed state because of a food effect. And we were also interested, therefore, to simulate the effect of PPI under fed state conditions. And we increased, therefore, the normal default pH value from 4.9 up to a value of 6.5. So applying these two modifications, we predicted actually a negligible effect of these changes on the PK of alectinib, which may seem surprising given the strong dependence of solubility on pH. However, when you consider how low was that aqueous solubility and that the dose was 600 milligrams, actually it's really a very small portion of the dose which could actually be dissolved in the stomach, and rather it's the enhancement of the solubility in the intestinal media, including bile salts and other components, which really is important to drive dissolution of alectinib. So that's really the explanation why there was no expected impact of this gastric elevation. This prediction was helpful at the stage it was made, because at that stage we were designing the phase 2 study, and we had to make the decision of whether to restrict the patients to those not taking acid-reducing agents. And based on this prediction, we didn't introduce that restriction, which was very helpful, in fact. Later on, as alectinib advanced, we actually performed a study with omeprazole, and that study confirmed the predictions of the GastroPlus modeling. So this is an example then of a molecule where PBBM indicates low risk of a pH-dependent DDI. And there are other examples of this. Perhaps most notably is another example from our paper, actually case study 7, which is ribociclib, a Novartis molecule. And in this case, PBBM simulations, together with POP-PK analysis of clinical data in patients, were able to support that there was no chance of a pH-dependent DDI for ribociclib. And so, in fact, there was no dedicated PPI clinical trial performed for this molecule. And if you're interested in that example in further detail, you will find a nice publication from the Novartis colleagues. Our paper includes at least 2 other case studies where we believe that PBBM could support very low risk of pH-dependent DDI. And again, in the supplement, you can find another example of perbenostat, which fulfills this condition of really a strong support for a lack of sensitivity to changes in gastric pH on the fraction absorbed. So that is summarizing this first group of studies where we believe PBBM could be used to waive clinical studies. In the second case study, which I will cover, is actually case study 3 from our paper. This is JNJX, a compound from J&J. And this study illustrates how PBBM can be very powerful at an earlier stage in a project to perform a risk evaluation. So JNJX is another weakly basic compound with which is lipophilic, and in this case it's BCS4 because the permeability is rather on the low to moderate side, and it's also given at or targeted to be dosed at a rather high dose of around 1 gram. This work was done to assess the risk early on in the development of JNJX, and what was performed was to run a 3D parameter sensitivity analysis in GastroPlus, varying the dose and the permeability. So I mentioned the permeability was on the low side here. And so you can see from these plots that if you look firstly on the left-hand side, this is at normal default healthy gastric pH in the fasted state. And so at this dose of 1,000 milligrams, you can see that the fraction absorbed is in fact shown here as a color coding. And the yellow indicates predicted fraction absorbed of around 60 to 70%. And this chart plot is also showing here that the permeability is really limiting the fraction absorbed, because as dose increases, we're moving to lower and lower areas of fraction absorbed. On the right-hand side, you see the same 3D PSA, but at a gastric pH of 5, and you can see that the colors have shifted because in this case, it's not just the permeability which is limiting here, but dissolution in the stomach also contributes. And so the fraction absorbed at a dose of 1,000 milligrams is reduced here to fall within this blue zone of around 30 to 40%. So clearly this molecule had a liability for pH-dependent DDI. Subsequent clinical studies confirmed, firstly, that the permeability limitation resulted in less than dose proportional increases in exposures. And later on, the molecule advanced, and a PPI study was performed and confirmed this predicted reduction in bioavailability at a dose of 1,000 milligrams. So I think a really nice case study from J&J here, showing how PBBM can be used from the early stage right through to later stages of development. Now moving to the last case study I'd like to cover. This is in our paper, the case study 6, and it concerns another oncology, Roche oncology drug, erlotinib, again for non-small cell lung cancer. So again, looking at the properties of erlotinib, you can see that it's also a lipophilic weak base with pH-dependent solubility, as you can see here. And again, you see here we had the measured biorelevant solubilities in the FASIV and FESIV media with which to build the models. Just a few more words about this case study. So this was actually done retrospectively. We had already the clinical studies with two different acid reducing agents, which were performed as part of the clinical development of erlotinib. But we really wanted to see whether the PBBM approach and the workflow outlined in our manuscript could be used in this case. So the first step here, and I'm going to go through the modeling in a little more detail in this case than I have in the previous examples. Of course, the first step is the description of the disposition pharmacokinetics. In this case of erlotinib, we had a study where erlotinib was given both IV and orally in a crossover design. And if you look at the plot on the right-hand side of this slide, you can see that quite clearly after the oral dose, the half-life is longer than after the IV dose. And so this is an indication of a nonlinear clearance. In fact, the use of PK plus was able to describe this data very nicely, and you can see that the parameters for a two-compartment model with nonlinear clearance were able to nicely fit these mean data from the clinical crossover study. Indeed, this model fitting emphasizes the importance of a good description of the disposition before we move into the modeling of the oral absorption, because by including that saturable clearance, we estimate bioavailability of erlotinib as 59%, whereas simply based on the areas, the dose normalized areas under the oral and IV curves, the bioavailability would be over 100%. So it's really important that we get this part of the model correct before moving further. So now we move to the description of the oral absorption using the ACAT model. And again, even after including this Vmax and Km, which we had estimated from the clinical data, and using that to simulate the oral profile, we still see an overestimation of the oral exposures when using the default ACAT model. And looking closely at the compartmental absorption, we see that the ACAT was simulating quite significant late absorption for erlotinib, so about 25% of the simulated absorption of a 150 milligram dose was predicted to come from the large intestine. However, that was overestimating the observed exposures here. Reducing the percentage fluid in the colon brought about a good match to the observed PK data. And so that model modification was included for further simulations of the effect of the acid reducing agents in the case of erlotinib. And now step 3, I come to the simulation of the effect of these 2 acid-reducing agents. The first study was done with the proton pump inhibitor omeprazole, and the second study was with the H2 receptor antagonist ranitidine. And so you see on the plots here on the right-hand side plots is the placebo, the data without acid-reducing agent, and then on the right-hand side after dosing. And you see for the simulation we used exactly the same adjustments with the increase of stomach pH to a value of 4. We also modified the gastric transit slightly because there are data supporting physiological changes in gastric transit, but in this case, these don't have a big impact on the outcome of these simulations. And you can see that this modification captures reasonably the observed effect, which was significant in both cases. What's also interesting is the plot on the bottom right-hand side, which is the sensitivity based on the model simulation of the ratio of the exposure with acid-reducing agents to that without. And you can see that this is extremely sensitive to small changes in the gastric pH within this range from 3 to just over 4 here. So summarizing then this case study, the PBBM modeling indicated here that there was a high risk of a pH-dependent DDI for erlotinib. I think if this was done prospectively, we would be challenging. It would be really challenging to make a confident prediction in this case. And that's for several reasons. Firstly, that there's this lastly mentioned high sensitivity to the exact gastric pH. And as we know, and as mentioned by Amitava, this is quite variable and depends also on the acid-reducing agent. However, the modeling for erlotinib was quite complex. We had the nonlinear pharmacokinetics. I didn't mention here that erlotinib is actually dosed as a hydrochloride salt, although the pH solubility data were for the free base. So that's another uncertainty. And there was also that uncertainty that I mentioned in the need to adjust the model for the colonic absorption. So it would be asking much to waive a study based on this modeling, given all these uncertainties. And we would recommend, and we recommend in our workflow in the paper, that for such cases, model verification with clinical data is recommended. However, if that clinical data does support the modeling, then certainly further use of the modeling can be envisaged, either to support further formulation changes or to make predictions in different patient populations with different extents of change in the gastric pH. So just summing up the presentation before we go into the questions. So we strongly believe that PBBM should play a key role in integrating physchem, in vitro, in vivo, and physiological data to yield fuller understanding of pH-dependent DDIs. Bottom-up approach and parameter sensitivity analysis is very useful early stages and to support internal decisions. There are many examples within our paper and also in other publications of the value to support clinical development. And we strongly believe that wider application of this type of modeling is supported, and that a waiver of unneeded clinical studies can be envisaged. And with that, once again, really would like to acknowledge our co-authors on this paper, They are listed here, and it was really a great pleasure to work with these colleagues and to pool our knowledge and come up with this nice paper as a part of the GastroPlus user group. And with that, I'd like to thank you and open up for the questions.
Okay, thanks again to Neil and Amitava for your presentation, and thanks everyone for attention. So now we are moving on to the Q&A session, and once again, you may send your written question using the question pane on your control panel, or if you would like to ask a question directly using your telephone, please use the hand raiser button on your control panel, and be sure to enter your unique audio PIN. So we have questions rolling in here. So the first question is, would particle size reduction help to reduce the magnitude of the pH-dependent DDI?
Sorry, I didn't get the first part of the question.
All right, so the audio may be a little bit garbled, but so the question is, would particle size reduction help to reduce the magnitude of the pH-dependent DDI?
I see. I would say it all depends on the specific case, whether it's really dissolution in the stomach, or is it simply the low solubility in the stomach, which is driving the DDI here. It may help in some cases, I would say, but I'm not sure it's going to help in many cases.
Yeah, and this is Amitava. I completely agree with that. In fact, we have one experience that we have published a couple of years back now, where it was primarily solubility, the low solubility, which was driving the DDI. And we did try nanoparticles and didn't help much with increasing the exposures.
Yeah,
okay,
so that's why I think PBBM is a key way to understand what's really driving this these effects, these DDIs, and that would help you to know whether it's worth investing in nanosizing, I guess.
Yeah, absolutely.
Okay, thank you both. So the second question is, does any anti-hypertensive drugs or anti-diabetes drugs have DDI with ARAs? I guess this is more of a question, from the clinical pharmacology side. So the question is, does any anti-hypertensive drugs or anti-diabetes drugs have DDIs with the ARAs?
Yeah,
I know oncology medicines are very subject to this type of effect, and as Roche is an oncology company, I'm more familiar with that area. I'm not so familiar with those other therapeutic areas. What about you, Amitava?
I would think, again, it comes down to the properties of the drug. I don't think we can generalize this based on an indication or a drug class per se. It will come down to the properties of the drug, primarily the pH solubility profile, and also I think we need to make sure that we understand that even if there is an effect of ARA on the PK, I think the key question is whether it is clinically relevant or not. So it comes down to, again, the physical property of the drug, and also safety margins and PK/PD of that molecule. So I think it will be very dependent on that molecule, and I just don't think we can generalize it by a drug class.
Yep, I totally agree with you, Amitava. So the next question is probably for Neil. In the Lieutenant case study, the GI transit time was changed in the model, will co-administration of the PPI-ARAs change the transit time?
Yes, it has been reported that PPIs can lengthen gastric emptying times. And in the case of alectinib also, we introduced that physiological change in the model. It had a small impact. It did have a small impact, but it was by no means a major impact. And it's I think we mentioned this in our paper. It's really much more the change in the gastric pH, which is driving these drug-drug interactions with these acid-reducing agents.
Thank you, Neil. The next question is more from the regulatory side. So the question is, will health authorities accept waiver of clinical study based on modeling simulation in the context of PPI-ARA interactions?
As mentioned with ribociclib, there is precedent for this, and another example where if you look at the FDA review for pabinostat, it is mentioned there also that the modeling work was supportive of no need to conduct a clinical study for those molecules. So I think this is one of the key points we wanted to make in certain cases where the PBBM is very solidly constructed and there are rigorous parameter sensitivity analyses performed to look at a wide range of change in gastric pH, and the model is really solidly built, then we believe there is a strong case here to avoid clinical studies, and it seems to be that agencies are open to that as well.
All right, thank you, Neil. So the next question is probably for you as well. It was mentioned that PBBM, PBPK, modeling with ARAs can be used to justify the change to the formulation composition and waive the bio study. Can you please elaborate more?
Yeah, so the first, what I just discussed was cases where modeling strongly supports a lack of a pH-dependent DDI. The second case could be examples such as erlotinib, which is the last case study I covered, where there is clearly an effect, and in that case we would recommend that the clinical data are used to verify the model. However, if the model could be verified and then a formulation change subsequently could be shown to have minor effect on dissolution based on in vitro data, then we believe that once verified, the model could help to waive additional studies with a modified formulation.
Thank you, Neil. The next question is, does any of the examples use the PBPK predictions to record a positive DDI in the label that and not doing a clinical DDI study? So I guess trying to understand the question a little better. So I guess this is more of a question. Yeah, still pretty much a waiver of the clinical DDIs.
Yeah, I think
with the PBPK.
Yeah, I think that's touching on the same point basically, and I would say I don't think there are such examples where just predicted on simulation no clinical study has been performed, and then in the label it's been recommended to avoid co-medications, which are acid-reducing agents. But yeah.
This Amitabh, I would agree with that too. To the best of our knowledge, I don't think there is an example yet of that.
Thanks both. So I guess just with the time limits, I think let's just go through another probably two more questions. There are many questions, but with limitation of the time, let's just get through the next two. So I guess one of them is that how does pH change in stomach influence the pH change in the small intestine where the drug is actually being dosed and absorbed? So I guess the question, if I rephrase it, is that if there is any impact, on the ARAs, the dosing of the ARAs to the intestinal pH.
Yeah. So I can start and let Amitabh add when he's heard my answer. So in the paper, none of our case studies included a modification in the intestinal pH. There are data to support that there are small changes in the upper intestinal pH with these acid-reducing agents, but it seems that those typically, again, similarly to what I said for the gastric emptying, those are less important than the change in the gastric pH, because generally we seem to be able to capture the changes with models which modify the gastric pH. However, I would say it couldn't be ruled out that if a compound is particularly sensitive to changes in the upper GI pH, then that may need to be considered also.
Yeah, so I'll just echo what Neil said. So there are publications there, at least the ones that I'm aware of, from Christos Repas's lab from Athens, where they have characterized upper small intestinal contents in achlorhydric subjects, and there is a change. But again, as to Neil's point, how much effect that has on the PK is probably not studied very well, and is probably not very significant, at least to the best of the information that we have right now.
Thanks both. So just with the time restraints, let's just get through the last question of this webinar. So the question is also more from the technical side, which is, will this model help in understanding the time for separation of administration of these interacting drugs? I guess this is more from the clinical practice side. Any of you would like to give a try?
Go ahead, Amitabh.
Sorry, Neil, didn't want to cut you off. I would say, again, I think if the model is properly verified against available clinical data, we could always simulate the time lag between the PPI and the drug. How much impact that would have on labeling obviously comes down to on the rigor of the model and how well it has been verified and how comfortable the regulators are with just studying it from a modeling perspective. Again, I'm sure I'm not answering this, but
if I could just add something, I think that's a very interesting question, and that goes a little beyond what we've shown here. I think that would need understanding of the time course of the the pH changes after a dose. And as we know, depending on the mechanism of these different acid-reducing agents, the time course of the pH changes will be quite different. For example, with a PPI, that I think would be a longer time course than for an antacid, for example. But I think that's really going beyond the modeling that we've described in this paper. Not to say it's not something which couldn't be studied in the future.
Thanks, both. I would probably just add a little bit about this question. I think that there are some reports and also in certain labels that actually does have a study of the duration of the interactions of the PPIs or H2 blockers that for certain drugs. Right? I think this is more of a case by case. Examples that so certainly that the question is an interesting question, but I would say that we will really have to look at it in a case-to-case basis on these. Do you agree?
Yes, I do.
Yeah.
Just a comment, Wei, just before we go. Maybe we could capture the remaining questions and we can try to provide some answers on the Gastro Plus user group webpage in the future if there are many questions which we don't have time to answer today.
Yep, sounds good. All right, so it is 6 minutes past 1 o'clock right now in the Eastern time. Thanks again to Neil and Amitava for your presentation, and thanks to Arlene from Simulations Plus for your technical support. So this concludes today's webinar. If you missed any part of today's presentation, the replay will be available at www.simulations-plus.com and the link in Gastro Plus user group page. And I would also encourage folks who doesn't get a chance to actually to get the answer of your question, post your question onto the LinkedIn Gastro Plus user group page. There are many questions, unfortunately, and there is a lot of interest, which is great on this topic. But unfortunately, we don't have that much time to cover everything. But please do feel free to post to the LinkedIn user group page. Again, and thanks everyone for joining us today.
Thank you.
Thank you. Bye.